In another experiment, they validate the clinical utility of CRISPR chip by using it to detect two mutations within the dystrophin gene that results in Duchenne muscular dystrophy. They use two DNRPs, which target two specific exons 3 or 51 in the dystrophin gene that are commonly deleted in patients with DMD. The results showed that CRISPR chips functionalized with DRMPs that were specific for exons 3 or 51 generated larger signal outputs after exposure to genomic samples containing the targeted exons, relative to DMD samples that carried the deletions of those exons. Based on the results of these experiments, it's safe to say that the technology is certainly one that has a lot of potential and is definitely viable. One of the advantages of CRISPR chip technology is that it is a small and compact system, which gives it the benefit of being the first point-of-care technology of its type. It's a simple three-step process: calibration, incubation, and rinsing that, if need be, could be done at a bedside if you have a good enough DNA sample. Many other methods for detection of nucleic acids require numerous reagents and large expensive instruments, meaning samples would have to be sent off to labs to be processed. Another thing that separates CRISPR chip from other field effect transistor-based biosensors is that it's one of the only ones that uses a whole clinical genomic DNA sequence. All similar technologies require synthetic oligonucleotides or very specific types of DNA. The authors don't explicitly mention any barriers with rolling out the technology, and it doesn't seem like there's anything that would drastically hinder the large-scale production of the product. The technology doesn't require expertise to understand the result and could likely be completed by doctors or nurses in hospital. The only thing that might present a problem could be obtaining the different guide RNA sequences for a specific genetic disease. You'd have to know exactly how and what part of the gene is affected by the disease, and this might be difficult to process for lesser known genetic disorders.